Idaho Matters is brought to you in part by St. Luke's Pharmacy, now offering free home delivery. For daily medications or one-time prescriptions, home delivery is easy to set up. It's convenient and safe. St. Luke's Pharmacy, now delivering to your doorstep. Information on how to get started at stlukesonline.org.
So you can't see this, but hopefully you can hear it. Behind every Boise State Public Radio podcast is a team working hard to bring you stories that matter. So if you value podcasts like this one, please support them. Hit donate at boisestatepublicradio.org. Thanks.
From the studios of Boise State Public Radio News, I'm Samantha Wright, filling in for Jimmy Gaudette. You're listening to Idaho Matters. For the past few hundred years, Rocky Mountain bighorn sheep in Idaho have been hit by everything from habitat loss to overhunting to deadly diseases from domestic sheep and goats. As their population and their landscape has shrunk, they now live in isolated pockets in the Salmon River region. And scientists are finding out that their genetic diversity has begun to shrink too, leaving them less able to adapt to the pressures that they face. Lisette Waits is a distinguished professor at the Department of Fish and Wildlife Sciences at the College of Natural Resources at the University of Idaho. Her Waits lab has been studying the genetics of everything from pygmy rabbits to red wolves to wolverines, even polar bears. She's part of a new study on Idaho's bighorn sheep, and she's here to tell us all about it. Lisette, thanks for coming back on the show.
Well, thanks for having me. It's good to be here to talk about our work today.
Absolutely. Well, let's talk about sheep. Which ones are we looking at today? I mentioned the Salmon River region. Where do these bighorn sheep live right now?
Well, they live throughout central Idaho and southern Idaho and the mountainous regions of the state. And, so I'll just end with that.
Okay, let's talk about the study. You used DNA to look at the genetics of the current sheep population. So how did you get DNA from today's sheep? I don't think you can just walk up and say, hey, can I have a sample?
Well, we got samples from a variety of ways. I mean, first of all, we're collaborating with Idaho Fish and Game in this project, and they manage the bighorn sheep, and they have collected samples for us over time. So sometimes they have captured sheep and collected blood or tissue samples. Sometimes if you can find a shed horn, we can drill out the horns and get DNA from the horns, or sometimes we collected fecal pellets and got DNA from that. So any kind of material that used to be part of the sheep, we can get DNA from.
Gotcha. And you went out and got quite a bit from today's sheep, and then you compared that to samples as far back as 1897. So where did you get those samples?
So we were fortunate to be able to have access to the Cary collection, and this was a private collection that's at the O'Connor Center in Lewiston, and this is a collection of bighorn sheep horns. And because DNA is pretty well preserved in those horns, we were able to drill into the horns and get shavings from the horns and use those to get the DNA that had been preserved there, and allows us to go back in time and understand what were the levels of genetic diversity and the patterns of genetic diversity in historic times.
So this is the John Cary collection, if I read right, and you looked at quite a few skulls or horns in the collection. There were a lot of them.
Yes, there were. There were hundreds in there, and we were able to get DNA out of quite a few of them.
All right. So is this a common thing? I thought this was really kind of inspired and a little weird to use basically old hunting trophies to do something like this.
Yeah, it's not common, and it's probably an untapped resource, but there aren't too many people who have such a large collection all in one place. Our lab has used museum specimens before to look at changes in genetic diversity over time in grizzly bears, for example. We've taken DNA from grizzly bear skulls. So museum collections in general are a great resource for being able to do this kind of study and go back in time and understand genetic diversity and patterns of structure in the past. But it's a lot less common to be able to collaborate with a private collection in this case for getting the DNA samples. So it is pretty unique.
Wow, and really helpful, I think. So you have the older DNA and the new DNA. So you compared them, and that got very complicated when I read your study. I kind of skimmed over that part because it got very sciencey. What did you find out when you compared the separate, I guess, time frames of DNA?
Yeah, so we found that genetic diversity did decline over time. So there were higher levels of genetic diversity in the past, and there was a bit more connectivity and gene flow between the bighorn sheep in the past. And one of the interesting things that we found is that there were a number of what we call desert bighorn genetic signatures in our genetic data. So we saw evidence that there had been gene flow and connectivity with the desert subspecies of bighorn, which would be found to the south of Idaho. And so that was an interesting finding. And we only found one sheep in the current sample that was showing that remnant DNA from the desert bighorn sheep. But we found, 30%, 39%, I think it was, of our historic specimens had this desert bighorn sheep genetic material. So that was a pretty surprising and interesting finding that told us that gene flow was a lot more common historically
So
with the desert bighorn.
yeah, so basically back then they were hanging out more with the desert sheep versus what they're doing now.
Yeah, and that there probably would have been a lot more bighorn sheep because that makes it easier for them to connect like in a stepping stone fashion across the landscape. But now there are big gaps, where we have roads and people and inhospitable habitat. But historically, it would have been easier for them to move around the landscape, and there would have been a lot more of them.
Yeah, and a lot of them now are kind of in real isolated pockets, basically
Exactly.
where nobody wants to be except the sheep.
Which is good for the sheep.
It is, it's harder to catch a cold from a domestic goat, I guess, if you're high on a cliff away from everybody else. So that's good. When you're looking at this is obviously a decline in genetic diversity. How bad is this decline? Can you quantify it?
Oh, we can quantify it. Like we can see the amount of decrease, and I would say it's a slight decrease in genetic variation. So it's not we didn't lose half the genetic variation, for example. We lost less than that. You can tell a significant drop, but we can hypothesize that that drop would lead to a loss of genetic variation that's important for adaptation, but it wasn't extreme. So in that sense, we're not as worried about these sheep in central Idaho. And these sheep in central Idaho are still exchanging genes with each other across that landscape of central Idaho. And that's a good thing for maintaining genetic variation and maintaining adaptive differences. So there's definitely an indication of decline, but not a cause for alarm, I would say.
Gotcha. Well, okay, you mentioned adaptation. So how important is genetic diversity to adaptation? Why does that matter? If they've got more diversity, does that mean they're more able to handle stuff that comes up like threats or diseases?
Yeah, you can think of genetic variation as like tools in a toolbox for, solving a problem. And so the more genetic variation that you have, the more ability that a species has to adapt to environmental change. So genetic diversity is the basic material for adaptation. So the more genetic variation that you have, the more adaptive capacity or ability that a species has. So it's super important. It is the most important thing for adapting to future change. And we know that the species in Idaho and across the world are being increasingly impacted by global change.
Gotcha. Okay, so you've got some results. What can we do with this information? How can this help the people who are trying to save sheep or make more sheep, I guess?
Yeah, so there's multiple ways. I mean, one, it shows how important genetic diversity is and how important this connectivity is for maintaining genetic diversity. So it's just another reminder of the importance of maintaining gene flow and connectivity across the landscape, because that will help us avoid losing more genetic diversity. It also, Idaho bighorn sheep were not extirpated like the California bighorn sheep, the Nevada bighorn sheep. The states around us, lost their bighorn sheep and they had to be reintroduced. Our populations in central Idaho were never extirpated. And so if we now know more about them, if they are going to potentially be used as a source of animals for other regions, or as we're thinking about do populations need augmentation or not, knowing their genetic variation helps us know whether they're at low levels of diversity and might need augmentation. In this case, we didn't find that, but that's one of the things we can learn with genetic diversity studies. So this provides guidance for potentially for future reintroduction efforts if we need to have any reintroduction or augmentation in Idaho, or if the Idaho animals might be used as a source for other states.
Wow, that's really cool. And I've got just a couple minutes left. I mentioned the Weights Lab. Tell us about the Weights Lab. What is it? What do you do?
Well, our goal is to use genetic methods to help answer questions for the conservation and management of wildlife and plant species. And so, as you mentioned at the beginning, we work on a number of endangered species because they have even greater challenges with genetics, and also because genetics can be a great way to track reintroductions. For example, we work on the reintroduced red wolf, the reintroduced Columbia Basin pygmy rabbit, the reintroduced Sonoran pronghorn, the reintroduced Mexican gray wolves, as an example. And we often use fecal DNA, fecal samples that we collect to track whether the animals that get released survive and how many of them are reproducing. And we also answer questions about mating systems and paternity and maternity in populations. And we answer questions about how the landscape is influencing movement and gene flow, basically trying to understand, are rivers or roads barriers to gene flow, and what things are helping maintain connectivity across the landscape. We've done that for wolverines, which helped us build a model for understanding how to maintain wolverine connectivity in the lower 48 states or across North America, for example.
Wow, fascinating. Well, thanks for sharing a little bit about your research and about the bighorn sheep.
Well, thanks. Yeah, thanks for inviting me. It's great to be on your show today.
We've been speaking with Lisette Weights, a distinguished professor at the Department of Fish and Wildlife Sciences at the College of Natural Resources at the University of Idaho, about a new study looking at the genetic diversity of bighorn sheep in Idaho.
And thanks so much for listening to Idaho Matters from Boise State Public Radio. Do you have thoughts about today's episode? Make sure you email us at idahomatters@boisestate.edu or reach out on our socials. And you can also listen to Boise State Public Radio podcasts, including Idaho Matters, on YouTube.
Every day on Up First, NPR's Golden Globe-nominated morning news podcast, we bring you 3 essential stories. At the heart of each story are questions. What really happened? What really mattered? What happens next? At NPR, we stand for your right to be curious and to follow the facts. Follow Up First wherever you get your podcasts and start your day knowing what matters and why.